it's another time for us all to get together and see what we can do for each other. And one of the things that you could do for us is join our speech study. As you're aware, research on ALS is continually progressing, and at Everything ALS, we are dedicated to uncovering new discoveries, aiding in early detection, and working towards finding a cure. That's what we want. We want that cure. And you can assist us by participating in our speech study. The purpose of the speech study is to enhance the diagnosis and prognosis of ALS. We are seeking participants to provide 5 recordings, each lasting 15 minutes once a month, for a total of 75 minutes over 5 months. We welcome healthy participants as well as individuals diagnosed or probable with ALS to join us. And if you'd like to enroll, everythingals.org/speech project. We also have one more project that's super important, and we would love to have your participation. We are actively recruiting for asymptomatic gene carrier. If you are a gene carrier and asymptomatic and interested in remotely monitoring your speech, cognition, and motor skills, please visit everythingals.org/asymptomatic and fill out the form. Your time is precious and you can make a big difference. So we welcome you to join us to cure this disease. Thank you all. And with that, I'll turn it over to you, Christian.
Thank you, McFinn, and welcome everybody. Thanks again for joining yet another one of our expert talks in our expert talk series, where it's really a great chance for you to be able to ask the experts in the field of ALS about both ongoing clinical trials and the other kinds of scientific topics that you might have questions about. So this is a chance for you to get straight to the point with them and ask your burning questions. Tonight, we're really lucky to have presenting CNM-Au8 survival, NFL biomarkers in a phase 3 update by representatives from Clene Nanomedicine. And joining us tonight is Rob Etherington, who is the president and chief executive officer at Clene Nanomedicine. Mr. Etherington has more than 30 years of experience in the pharmaceutical industry, and Rob's led Clene's focus on ALS and MS for over a decade. Prior to joining Clene as CEO, he worked for Actelion Pharmaceuticals, which was the largest biopharmaceutical company based in the EU until Johnson & Johnson acquired the company in 2017. Prior to Actelion, he served in a number of roles at Parke-Davis, a division of Pfizer, and culminated in a team leader position that oversaw the drug Lipitor. As the founding CEO of Clene, Rob has raised more than $200 million in financings and has driven Clene's focus on neurodegenerative diseases. Also joining from the Clene team tonight is MarjanSepassi, Vice President of Medical Affairs. And Marjan brings over 15 years of clinical, medical strategy, and health economics and outcomes research and team leadership experience to Clene Nanomedicine. Prior to joining Clene as the VP of Medical Affairs, Marjan led medical strategy and field teams at Civi Biopharma, Arena Pharmaceuticals, which is now part of Pfizer, and Actelion, which is now part of J&J, in rare disease areas of high unmet need. Marjan's entry into biotech started as an academic researcher focused on hematology oncology. She holds a Bachelor of Science in Molecular Cellular Biology and a Doctor of Pharmacy degree from the University of Arizona. And tonight they're going to be presenting a really important update on their phase 3 trial. And then from there, we'll open it up to some Q&A. So at this point, we're going to turn it over to Rob and Marjan.
Thank you, Christian. I just realized we need a send request for multiple presenter sharing.
Absolutely. So
great. Thank you. It is a pleasure for Marjan and I to be with all of you tonight, and we're thankful for you taking some time, and thank you, Christian, for that nice introduction. So you've already heard a little bit of our bio. So what we're going to do is Marjan and I will go back and forth for the next 20 minutes or so, talking about our asset that many of you might be familiar with, and then we'll take time for question and answers. So the way we're structuring this presentation is we're going to have Marjan talk about the mechanism of action. give a little bit more, clarity as to how an oral nanosuspension that I'm holding here in my hand, this is actually the investigational product, a sample of what patients are drinking every morning. People with ALS are drinking every morning in our clinical programs and in our expanded access protocols. Then I'll talk about some of the clinical data that we have shown to the agency recently and where we're going with that. And then Marjan will talk about the planned phase 3 study. So let's turn now to Marjan to talk about mechanism. Marjan, you're on mute.
All right, it worked this time. Thank you all for joining us today. It's a pleasure to be here. It feels like we were just here just yesterday, but it has been several months. We had questions last time about mechanism of action, so I thought we would take a few minutes to talk about ALS in general. We know that in ALS, motor neurons die in various ways. We know this through familial ALS, through sporadic disease, where you've got variability in the progression and the death to the neurons. Now, regardless of the insults that you see, whether it's toxic proteins, oxidative stress, whether it's cytotoxicity, all of these things ultimately converge and are a critical driving force to the neuron ultimately deciding to die. Next slide. So I won't take much time other than to really show you guys the nanocrystal CNM-Au8. It's an oral suspension that's in a 60 milligram dose. It's orally administered or through a feeding tube. It targets energy metabolism and oxidative stress. It crosses the blood-brain barrier very, very nicely. We've shown that via MRI spectroscopy. We've not done it in ALS patients because with an MRI machine, we have to have folks lay down for a long period of time. But we've been able to show target engagement and crossing of the blood-brain barrier very nicely in individuals with Parkinson's and MS. And the bottle is shown right nicely there on the screen. It kind of looks like a nice, a shot of wine. It tastes just like water. In fact, as Rob has talked about previously, we have to make sure that the taste matches the placebo because by and large, it tastes like a really, really nice bottle of Evian water. And we can move forward. Thank you. So essentially, what is CNM-Au8 and how does it work? It's essentially it works via promoting catalysis. So we have a natural system within our bodies that is catalysis. And all we do, CNM-Au8 is not a molecule that's inhibiting proteins. It is not a gene therapy. It is not cell therapy. It doesn't require cellular machinery or any kind of energetic input. All it simply does, it permeates cells and provides an environment where NADH is naturally via catalysis able to promote the conversion of NADH to NAD and improve mitochondrial function and ultimately take the neuron with it. And with that, we get the mitochondrial health and survival, and we do the same with the neuron.
So now I'll take over and give some clinical sense. So just many of you even maybe are familiar, but not everybody. So we've run 3 collective programs in ALS. The first is Rescue, a 9-month study of 45 participants that was entirely conducted in Australia and funded by the Fight Motor Neuron Disease team there, for which we're super grateful. The second was the Healy program with Merit Sudovich and her team at Mass General and at Harvard, and funded in part by the Healy program and the balance from Clene. And that was 161 participants over 24 weeks. We were regimen C. In both the case of Rescue and in Healy, we continue to treat many people in the open label extension. And then we have 3 expanded access protocols, sometimes called compassionate use, but definitively spoken to as expanded access protocols The first one was commenced in the fall, winter of 2019. And that was a combined project between Mass General, Harvard, and ourselves. And this was designed for individuals that could not meet the Healy criteria and wanted to continue on compassionate use nonetheless. The second is EAP02, and this one's funded entirely by Clene. And the third is the NIH program that commenced last June, so basically 8 months ago, from the money that Congress allocated to the National Institute of Health and funded. We're now in year 2 of that funding, beginning in 25. And that program fully enrolled at the present group in late year, December, in fact, of 2024. And so taken together, there's 477 participants that have been treated in our expanded access protocol, so quite a few. And you see some check marks on this page. All both of our programs, as well as all of the entirety of the Healy programs, have missed the primary endpoint. And in our case, it was Munich's in the case of rescue and ALS-FRS in the case of Healy. But very gratifying to us, and what has kept very much us working day and night, is we saw delayed time to clinical worsening most definitively in the rescue program. And subsequently in our open label, we've seen survival and preservation of function. And then in Healy in particular, we saw survival as a key secondary for the 30 milligram dose. And we also saw as the double-blind placebo control achievement of delayed time to clinical worsening. And we also saw a p-value that was statistically significant in neurofilament. And as you see from the open label experience, that continues to actually grow robustly, including other biomarkers. And so let's talk a little bit of context of this. But first, I'm going to invert what is normally done in biotech presentations. Usually, we talk about safety last. We talk about safety first in our case because, frankly, there's no safety issues. There's no story behind safety. It's actually a definitive safety argument that has been able to be expressed by Clene. Very gratefully, knock on wood, as it were, super grateful that we have not had a serious adverse event that has been attributed to drug in any of our programs to date. So considering ALS and expanded access protocol participants who are too sick often to come into a clinical program or for various reasons choose not to, but also multiple sclerosis and Parkinson's, all of these have 0 related SAEs. And this is a very big deal in our pharmaceutical world. An SAE stands for serious adverse event. It's something that the doctor attributes happening to the patient or participant in a clinical study because of the drug. And normally our world revolves entirely around safety concerns. But in Clene's case, we have some safety issues. You see here as the third bullet, treatment emergent adverse events that include occasionally headaches, sometimes nausea, sometimes some diarrhea. But actually, the longer you're on the drug, the lower these incidences are. And even as it is, we have very, very few, such that you can count them respectively on fingers and toes, of people who complain about these programs such that they can't take the drug. In other words, we have no adverse event findings really across any of our programs with CNM-Au8. So we're super grateful by that, because one of the reasons, incidentally, we call the company Clene is we theorized at the beginning without chemistry, and what Marjan just spoke of has no synthetic chemistry whatsoever in our production of this asset, that we would potentially have a clean toxicity profile, that is safety that was strong. And we're grateful that we do now across collectively 800 participant years of exposure. And we have participants in these EAPs and in the Healy OLE and in rescue that have now been on drugs since the winter of 2019. So that's now quite long, 5+ years. Okay. So let me talk about neurofilament. You all know this quite a bit, and it is something that's very important for Clene right now because it is the key path that the FDA has suggested is a surrogate opportunity for us with respect to understanding the potential of submitting a new drug application. So I think you're pretty familiar, but just to make sure, as the disease ALS attacks the neuron, and there are other neurofilament examples and other neurodegenerative diseases as well, but we'll stay focused on ALS. Then what actually results after that attack is a cytoskeletal protein that can be measured in the blood. And so there are other such examples of biomarkers. I was talking to somebody way less experienced than many of you a few hours ago, and he made the point. So in a way, glucose, if I'm diabetic, or cholesterol, if I'm an atherosclerotic cardiovascular compromised patient, or even if I'm a guy, my prostate-specific antigen. All of those are biomarkers. Neurofilament is one such example, clearly. And a high neurofilament in ALS predicts quite evidently axonal damage as well as disease progression. And we saw on the left in Healy statistically significant during the 6-month period, double-blind, statistically significant relevant difference in neurofilament. And that's the green line. The placebo patients are in gray. And what's interesting on the right is during the open label, we saw that event, that neurofilament achievement, become even more robust, about a 70% improvement, as you see here, in terms of a geometric mean ratio difference. And what's also fascinating to us is in the Healy program, when the 6-month double-blind program stopped, the vast majority of participants chose to proceed on to active open-label drug. So after their placebo period concluded, we had some 90% of patients that moved into the active label, and we continued to give active label to now both groups, the original active and the placebo patients. And what is represented here in green is about the time it takes to get to steady state blood levels of CNM-Au8 within the system. And once that occurs, we seem to see a stabilization of the neurofilament increase and then a drop once we have active drug on board. So the 70% improvement is even after we're seeing the original placebo people, the people with ALS that had placebo originally, they start to drop in their neurofilament as well, and it's about a similar slope as the original actives. So we took this data into the FDA, and the FDA asked us to look at things quite comprehensively when we presented it to them in late November, and we presented this to the public in a press release with subsequent slides, which I'm about to show you, when we received the full minutes from the FDA. And we talked to the FDA about it like this. First of all, to look at plasma neurofilament burden at baseline on this road to time to death, or PAV, permanently assisted ventilation, which in ALS clinical studies is really best defined as when I need 22 hours a day or more of permanently assisted ventilation. And then secondly, neurofilament decline. So let's first look at this first. So this is looking at neurofilament at baseline on the left, the upper tertile. So if I take neurofilament and I just look at it in thirds, and the worst third, the highest neurofilament for the top third of participants in the Healy program, we see this curve with respect to risk reduction. And this is, it's a horrible word that I don't like, time to death, or PAV. I like to think about it, those who are living, those who are surviving. And we see an 83% risk reduction with a p-value of 0.03 here. The gray is placebo, the active drug is the green. So what this shows is interesting. So if my neurofilament is particularly bad, then I have an interesting survival benefit that is quite robust. On the right, I'm looking at everybody who's just above the median. So basically the top half. So it's the third top half, and both numbers are concordant. So an 83 and 84% risk reduction, both significant, at 12 months of being on the drug. So if I'm on CNM-Au8 and I have a high neurofilament that's above the third, or at the top third or the top half, in both cases, I have a nice hazard ratio, which means an 84% risk reduction, which means a less likelihood of passing away. And so then what we did is we wanted to look at all decliners, just look at neurofilament declines. So for all subjects on AU8 who had a neurofilament decline, we see that that's about twice the number of individuals that were on placebo with declining. And let's just look at those decliners. Let's look at those that went on drug and had a reduction in their neurofilament. And that's the red box and everybody below the red box. So any decline whatsoever of any measurement. And what we see here is actually, if we just look at decliners, we have a stronger risk reduction in terms of my time to possible of passing, and a p-value of 0.001. So pretty interesting data suggesting pretty nicely that neurofilament, in the case of CNM-Au8, if I enter a program in AU8 with a high neurofilament and I can reduce it, there is a benefit that occurs to me with respect to survival. And then we actually, and that presentation was given, by the way, by the extraordinary Dr. Bob Bowser, who is one of the world's top neurofilament experts when he presented to the agency. And then Dr. Merit Sudovich also joined us, and Dr. Jinsey Andrews, both extraordinarily amazing clinicians, for which we are, frankly, immensely grateful. And Dr. Merit Sudovich talked about clinical trial populations and some work that we had done to take her extensive, her and many others, extensive databases of pool match controls. Now, this is coming from PROACT, the ALS Natural History data set, and ANSWER. So 3 different data sets that have been collected by the work of many people over many years to really understand natural history of ALS. And this is super relevant because, I, like you, would want to be on placebo as little as possible. And so what it does when we are following these participants in these programs from these clinical studies, and the left HEALEY on the right, HEALEY and rescue together, it's challenging if we don't have a placebo comparator set, which we don't, because in these 2 cases, after 6 and 9 months, everybody naturally moved over as fast as they could to active drug. But if we use the most conservative matching we can find, which is a statistical method called greedy nearest neighbor match, just as an aside, and we do what's called propensity matching and covariate adjusting. So basically, said a different way, we're looking for an apple-to-apple comparison. We want like patients that are matched across a number of covariates, similar gender, similar SVC, slow vital capacity, similar ALSFRS, and so forth and so on. And then we look at those individuals in a statistical model. We found that here we have a p-value of 0.2 and 0.1, respectively with HEALEY alone or with HEALEY and rescue, that both have similar, or in statistical words, nearly concordant risk reductions over a long time. These are programs that we're tracking these participants for up to 4 years, actually. And we see this survival benefit continue to stay kind of consistent across the clinical program. And then what we did is looked at the same analysis with our EAP participants, and we see the same thing, another 30-odd% risk reduction that in this case was even more highly significant over 4 years plus. I mentioned already that we've had people in the compassionate use EAP programs now for over 5 years. And this gives you some example of what I mean by covariates. Again, I'm matching by baseline BMI, baseline gender, baseline bulbar onset, whether it's bulbar or limb onset ALS, baseline months from symptom, my onset age, my diagnostic delay, how long it took a doctor to get me diagnosed before I could start, getting at this disease, my pre-treatment ALSFRS slope, my total score, how well I'm breathing, and also my TRICALS risk score. So taken together, all of this is matched, and we find that we have a pretty solid hazard ratio, which means to say a nice risk reduction in my possibility of passing away. And so this is all remarkably important data for us. We're continuing to interrogate this data quite exhaustively on survival because we're continuing to track these patients. We have now, this is all data from summer of last year. So we're coming on another year of being able to leverage this data. And I should also add, jumping back, the reason why this neurofilament argument becomes so important is, as Clene has announced, the FDA asked us to use the neurofilament understanding from our EAP compassionate use that has been funded by the NIH. So just a little comment about that. In the NIH program, when NIH approved the protocol, neurofilament collection by home-based phlebotomists who come on site to the person in the program's home so that the collection is easier, is then that blood is taken every 6 to 12 weeks, depending upon where I am in the protocol, and frozen, and it's all sitting in a central laboratory. Hasn't been looked at. Neurofilament in clinical studies needs to be looked at once when a program concludes. And later on this summer, we will have matured that data to enough number of people over enough time to be able to interrogate that data and to see with the EAP if we're seeing concordant, that is similar, arguments on neurofilament reduction and to get some early sense if it's tied to survival. So we're very excited about that opportunity because if that is the case, then we plan to submit this data to the agency and ask for their consideration of the possibility of a new drug application. Taken together, the survival arguments you've seen as well as the neurofilament arguments you've seen. Now, in the midst of all of that, we would not even be considered an applicant for a new drug application for accelerated approval unless and until we commence a phase 3 confirmatory clinical study. So let me turn to Marjan now to talk about our plans for this.
Thank you. So Rob did a great precursor to really the reasons that we have to believe and why we're here today is ultimately moving forward with CNM-Au8 and discussions with regulatory bodies and hopefully having approval, but at the same time, conducting the phase 3 program. At the moment, I think I've spoken about this several times, but I'd like to review it for the team. Phase 3 is called Restore ALS. It's a randomized, double-blind, placebo-controlled trial in early symptomatic participants on stable background therapy. I'll talk about that a little bit later. It's one of the differentiating factors that we think makes the Restore program hopefully more palatable for those who would be interested. So it's for those who are unstable therapy to reduce mortality and obviously clinical worsening events in those diagnosed with ALS. We will be starting in the Asia Pacific region. We have great experience conducting our phase 2 rescue program there. Getting programs up and running in that region tends to be a bit faster. So we'll be starting with Asia Pacific, specifically Australia, moving towards Europe and eventually North America. So as Rob alluded to, and our reasons to believe, survival is one of the key endpoints that we were able to see from the Healy platform trial. And as you know, as you take those, bits and pieces of data that you have most effect on forward into a phase 3 program. So we're looking to investigate the improved survival in people living with ALS. The program is a 108-week double-blind study in duration in order to make as many folks take active product as possible. There is a 2-to-1 randomization. The double-blind period ends at the accrual of 220 events. Essentially, what we're doing is a time-to-event trial. So once we get to 220 events, the program would end, and we would have the safety follow-up period, as well as if we were to get to 108 weeks. So this is a longer program. One of the ways that we've thought to make it more palatable and have it be a program that people are committed to and stay in is the ability for optional remote visits. So after you go through the initial baseline visit and week 12 and week 24 visits, every other visit has the opportunity to be optionally remote. And then in the end, we also have a little bit more flexibility as well. I think it's important to note that we do plan to have interim analysis for futility. And essentially that means that once we collect 50% of our data or 75% of our data, a independent committee termed the DSMB committee will review the data and see if there's any potential harm or if the investigational product is not working. That way we can limit the exposure that participants have to a product that may not be working and be able to terminate study early on. Now, that could happen in reverse, which is what we hope as well. If the DSMB warrants that everything is going well, we're obviously going to continue. Why is survival as a primary outcome? As I mentioned, from our phase 2 program, survival was our strongest signal, and that is what we'd like to have moving forward. It's also a signal that's very important to regulatory bodies, both here in the US and across the globe. It's something that still regulatory bodies insist on and want data on, in spite of our movements with ALSFRS-R and other functional endpoints. As well as we ourselves did as we started to plan the program, we did a survey of the community. And again, from those people that are living with ALS, this is a survey that was sent out by the Synapticure team. Survival is still something anybody living with ALS considers very, very important amongst functional outcomes. So as I mentioned, the primary endpoint is time to death, or PAV. PAV is defined as mechanical or non-invasive ventilatory support greater than or equal to 22 hours per day over a 7-day period. We're also, as a key secondary endpoint, looking at composite of ALS clinical worsening events, essentially as a time to death PAV, need for tracheostomy, need for dependency within 3 regions of the upper 3 regions. That means ambulation, speech, upper extremities, need for feeding tube placement, need for assisted ventilation, or none of those. We're also looking at joint rank scores in various endpoints as part of our secondary. As I mentioned earlier, the 2-to-1 randomization assignment essentially means that we will hopefully have approximately 460 or so individuals that will be on active treatment, with about half on matched placebo, with a total of 690 participants being randomized. This is a larger program. As I mentioned again earlier, we do allow concomitant medication therapy. We do want it to become more palatable and move with advancing science. So currently approved therapies will be allowed on as long as they're at a stable dose based on local approvals within the country of enrollment. We will also allow for newly approved products that are that finish their phase 3 program on our approved during the course of the phase 3. We will also allow newly, investigational products that have positive outcomes via a phase 3 program that will go through that independent data safety monitoring board that I mentioned earlier. And based on those results, they will determine whether we can include it in the program as well. And then I'll hand it to Rob just to kind of talk a little bit about our overall priorities, or where we stand in terms of our discussions with regulatory bodies. And then I think we'll answer questions.
The rescue is effectively concluded. There was a question in the chat here. Dr. Stephen Vucek and Dr. Matthew Kiernan are the 2 key ALS doctors in the country of Australia who did this program. And Dr. Steve Vucek in particular will plan to be the PI of the Restore launch. Dr. Healy, of course, was led by Dr. James Berry and Dr. Merritt. And the expanded access programs, all three of them noted here, are all in our responsibility. And then the plan Restore is coming up. So where we are is we're just initiating regulatory engagement in Europe. It'll be a little ways there. We expect the Europeans will primarily wait for Restore. And in the case of the US FDA, well, that remains to be seen. I think that the US FDA wants to make sure that there is, to much degree as they can, that there is not a false positive in any of the data we've shown them to date, which is one of the reasons why we thought it was a clever proposal to lever the 180 subjects from the EAP. There was a question in the chat about the SynaptiCure program. All of those participants in the EAP are on active drug, just to be very crystal clear. The only placebo programs are already concluded, Rescue and Healy, and those individuals who were on those placebos were invited to come to active drug if they so choose, and we continue to treat those individuals in the open label extension. So what Clene is now preparing for is the maturity of enough subjects in the Synapticure/Columbia/NIH EAP, we call it officially EAP04, to actually reach the time where they're on drug at least 6 months and beyond. Because in Clene's case, it takes a moment to work. And so we're wanting to make certain we have that evidence. And then the FDA asked us to come back and speak to them in the second quarter before any of this analysis occurs to have an alignment between the statistical analysis plan. So to show them exactly how we plan to evaluate this data and to have it, if you will, pre-specified with them on its statistical analysis before any of the participants' blood samples are thawed and analyzed, which all has to happen at the exact same time. So that event of analysis will be, again, somewhere in the summer period, and then that data will be sent back to the FDA. So with that, Christian, let's maybe turn to questions. I see a number in the chat. Some we've tried to answer throughout, others are new.
Thank you so much. That was a action-packed presentation, a lot to it. So, and a lot of great questions came in. So, first and foremost, since it wasn't covered too much in the presentation itself, is there still any opportunity to get in on any of the EAPs? And are there any potential 2025-26 funding impacts to Clene SynaptiCure EAP on your radar?
You want to address that, Marjan?
Yeah, sure. So as Rob talked about, we have several EAP programs, and yes, we didn't talk about EAP, but I know it's always top of mind for everybody that's in the community because that is one of the only avenues at the moment to have access to CNM-Au8. So while we try to maximize and provide as much exposure to those who don't qualify for traditional clinical trials, exposure to CNM-Au8, at the moment, the EAP programs are essentially full. That includes our program through Mass General, our second EAP program that started post the phase 2 programs, as well as the NIH program. If you guys recall, the last time we were here, we talked about expansion of the expanded access program where we were able to reallocate some of the funds that we received from the NIH to enroll additional people living with ALS interested in the expanded access program. We did do that, and obviously that filled up very quickly. It is definitely something that continues to be on our radar. We've had discussions with the NIH. They too are open to the potential, but at this time, do we have that approval? No. One of my suggestions would be, please don't stop reaching out to us as a company, as well as SynaptiCure or any of the brick and mortar sites that are across the country, because we are keeping an active list of people that are interested for the expanded access program. So should we be able to implement, and I'm crossing my fingers and toes that we are able to expand a little bit more, that we will reach out to those based on, SynaptiCure has their own model, as I talked about before, as well as the brick and mortar, but we're hoping that we could. In terms of funding, obviously, the NIH funding is technically an approval for a 4-year time frame. We did get the first year funding, and I'm happy to share that we have gotten the second year funding. The political landscape was one of the questions. As far as we know, it was approved for 4 years, and we have received the second year of our funding. So as far as we can see, that the program would continue, and we would receive our additional funding.
And we should add that second year funding was signed by the NIH in the last 30 days. So that's after some of this drama has commenced, which we hear about obviously in headlines. And so we are under no knowledge that there is any impact whatsoever to ALS patients. And of course, it would be extraordinary if there was, and Clene would fight tooth and nail to advocate for you as we do every day. We are grateful for everybody we have been able to help so far in the EAP. We'd love to be able to help more. In fact, the decisions made by the clinician, but we do, of course, allocate investor capital to this, and it is presently a large part of our budget, even in these difficult raising money for small pre-revenue biotech company times. So we certainly appreciate all of you that have participated in these programs.
Thank you.
Awesome. Thank you guys so much for answering that question. Moving on again with EAP, why were the functional outcomes in EAP not assessed?
So go ahead, Robin.
Go ahead, Marjan.
So, overall, what is the scope of an EAP? Maybe I should start by saying that's not entirely true, right? By and large, yes, expanded access programs or compassionate use, as we all term them, are meant to provide access. So funds are allocated, and we try to maximize the number of slots that we're able to give those interested. So it's really about balancing the dollar spend and the program infrastructure to the number of individuals that we're able to give EAP access or compassionate use access to. However, for the NIH program, that statement isn't true, because for the NIH program, not only are we looking at survival and safety, survival obviously being a key endpoint for us regarding CNM-Au8, but we're also looking at clinical functional outcomes like ALSFRS-R. We're looking at changes in clinical care. We're looking at changes in fatigue. We're looking at changes in breathing via FVC. We're also looking at biomarkers and quality of life measures. So, one of the great things that the NIH grant has done is really provide the opportunity to use funding to move the needle as it pertains to real-world evidence generation in an expanded access scenario, where we never get that from a phase 3 program. A phase 3 program is a very small sliver of what we know about CNM-Au8, or a phase 2 program, which is designed to be a dose-finding study. So this is real-world evidence generation, and I think there's a lot of interest to see how these programs, yes, that utilize a lot of funding, that utilize a lot of infrastructure at the same time, we're able to generate evidence that's supportive and provides, additional information from the public that we normally wouldn't get. And Rob, I think maybe you had something to add for that.
Yeah, I was just going to say, if we added functional to all of them, we'd be able to treat less people in total, because the functional assessment mostly is a clinical cost, and it has to be done relatively soon. So we've saved the function assessments primarily for the clinical studies, but as you've certainly seen from Restore, the phase 3 that's just been proposed, time to event, that is how many people we can keep alive on Au8, is our primary endpoint. Next question.
Thank you.
Yeah. So is there a dose response signal in the trials or EAP? And if there is no dose limiting toxicity, then would a higher dose be worth evaluating?
Two answers to that question. We actually saw in 60, and if you read the JAMA paper, you saw that we did only 30 milligram in all of our MS work. We did only 30 milligram in Parkinson's. We did only 30 milligram in rescue, which is the Australian study. When we were speaking to the agency, that same question was raised. Might there be a dose response? And so we put into the Healy program a 60 milligram dose, which frankly is quite difficult to manufacture. That actually takes us a lot more time and money to pull the 60 off. But we saw, very interesting to us and kind of different than we would expect in neurology studies of drugs normally, that the 60 milligrams showed no benefit. We did not see a 60 milligram survival benefit, for example, and the 30 milligram benefit was also primarily in the neurofilament. So it seems as if 30 milligram, where we were with all the rest of our programs from the animal studies at 30 milligram, was in fact at the top of the dosing range. And so you are correct that 60 didn't show any more side effects from the Healy trial, but it did not show any benefit, and it is quite more complicated to make. It takes twice as much time, twice as much money, and twice as much drug, frankly, for us to make the 60. So we have anchored 30 as our top dose, and there is actually a fair amount of mechanistic rationale for that in the way that a nanotherapeutic works within the body as well. So 30 is the top dose.
Thank you. I'm going to skip ahead. I know we've got it. We take questions usually in order, but I'm going to skip around a little bit here because I think we've covered some of the functional questions as well. What would be the effect of CNM-Au8 in the phenoconversion of ALS when the compound will be given symptomatically or pre-symptomatically to prevent phenoconversion? Is there a possibility of that?
Good question, and I don't think we have enough data to know. We consider CNM-Au8 as an add-on therapy. As far as the data that I'm familiar, I don't think we know enough. Rob, would you agree?
Yeah, that's true. We've not done phenoconversion studies, so that's a good question in terms of kind of if ALS is, as it were, coming or just beginning. Rescue, just as an aside of that, rescue individuals were only 4 months diagnosed, so they were pretty early in their disease. They were, I would argue, at 4 months diagnosis, very early. Healy was classical 6 to 12 months diagnosed, so they were quite different advanced. But the phenoconversion study aspect of this, we have not actually explored other than what we might have learned from rescue at Healy, which was not much.
Thank you. Did you want to take the next?
Yeah. Data suggest delay in effect for 12 months. Are slower progressors more favorably affected?
Marjan, you want to take that?
No, you can go ahead. No problem.
No. So that is a good question. That's a very important question that we don't actually know. So we're trying to figure that out because it's very important for Restore, the phase 3. In fact, another question in here is there a minimum neurofilament level that one needs to have to come into the study? And we are right now talking with the clinicians and those that are guiding the steering committee of Restore to have just that, a minimum threshold by which a patient needs to have on the neurofilament baseline to enter. That is the most likely that, as you saw already from the survival data presented, the worst neurofilament patients have the highest survival benefit. So it behooves us as we design this inclusion criteria to have such. Now, how to discern slow or fast progressors is a conundrum that we're also right now doing statistical modeling on as we wrap up the last piece of our protocol. Marjan, any other comments on that?
Yeah, I think to be a little bit more precise, we hope, and at least the current design says that we will have a, NFL baseline that will be greater than or equal to 45 picograms per ml. And essentially, like Rob said, we're trying to be right in the middle. We want to avoid really fast progressors who won't even have the time on therapy to get a therapeutic effect. We also know that CNM-Au8 has a very long half-life. The half-life is almost 1 month. So in order to see that therapeutic effect, we need to have those that are taking the active product on therapy for an extended period of time. So that's one thing to consider. On the other side, we want to avoid really slow progressors that would not contribute to the primary endpoint, right? In the end, we want to finish a phase 3 program.
Thank you. going to jump ahead again. What do you think is different about the neurofilament non-responders? Do you have thoughts about a predictive biomarker that would identify these people so we would know who is most likely to respond?
Marjan, please.
Yeah, I mean, that's kind of what you're trying to do with having that minimum threshold, right? You want to have that sliver of individuals for a phase 3 program that we think are most likely to see that effect. And we do that not just with the NFL values, we do it with, the SVC levels. Like, for example, I think we would need to have, at least with our current design, an SVC level that's greater than or equal to 60% of the predicted. Or we would do the TRICALS risk score. We have a range that is appropriate for our program of -6.5 to -2.5. And that really is that mid-range to maximize the number of individuals that we believe would have the effect on CNM-Au8.
And I should also add, there are many other biomarkers that are of research interest. We've been talking to Dr. Bob Bowser about these. In fact, we're thinking of actually paying for 2 different assessments of biomarker understandings in the Restore protocol. There is a group in San Diego called Sapien that uses this new technology called Alamar that, unlike Quanterix that just has neurofilament only, they have up to 120 different biomarkers that researchers are still trying to understand if they have merit. And so we're trying to be as exhaustive as the dollars would permit to look at multiple biomarkers beyond neurofilament. A little bit the wrinkle is that neurofilament is what the US FDA understands the most at the minute. Now, Biogen pioneered that. They've done an extraordinarily good job of helping the FDA understand neurofilament. I was stunned because I was just at the Amsterdam-based TRICALS meeting of many European key opinion leaders, and neurofilament still remains the primary theme that interweaves almost every discussion there. Other biomarkers are rarely discussed. So we still have some ways to go, I think, in the academic community and the research community to understand ALS biomarkers, but we're trying to make as much progress as we can.
So a quick follow-up on that. Presumably then, do you think there is a subset of ALS patients or a stage of ALS in whom the mechanism is more primary? Could it be used that way? Because you saw people who clearly showed an NFL response and others who didn't. So there's a presumed mechanism.
Yes. And that is actually one of the reasons to point Marjan just made, that we're looking also for neurofilament cutoff, because we want to see where the neurofilament is, and then we want to understand how neurofilament changes over time as well. So we agree. Yeah.
Move on to the next question. What type of subset or subsets were non-responders to the drug?
What type of subsets were non-responders? I don't have any good Healy program. It wasn't addressed really in other words, you're basically asking, if we take Au8 and we don't have a survival or neurofilament benefit, what is the reason why? And we've not yet been able to interrogate the database sufficiently to understand that question. Hopefully Restore gives us more of a sense.
So actually, I'm gonna skip up ahead. There were some really good ones earlier on as well. We talked a little bit about functional. Did you see effect over time on FVC or atrophy or any of the other core ALS FRS as you were tracking progression?
Yes. Yeah, we did see over time a trend in the right direction. So, one of the things that we learned from the phase 2 programs was time is very important, right? Whether it is the program that we had in Australia or the Healy platform trial, especially for CNM-Au8, we need to be on therapy for an extended period of time past 6 months to start to see that. But in those programs, we did start to see a trend in the right direction.
Okay. And also from results, could you please tell us the probability, this goes back to the graphs you were showing too, the probability of false positives. In most of the matched control studies, it was visible that the placebo numbers were high. What if the placebos are too fast progressor? Then there would be a chance of false positives with effect in the treatment group.
I mean, there's always that chance, but you hope with I mean, that's why we do a phase 3 program, right? That you minimize those confounding factors to an absolute baseline minimum. Yes, it's always possible, but to the best of our ability, that's why we do a phase 3 program.
And I would add that's exactly the question the agency's raising. So that's exactly why, just to put this into perspective, of the rescue study, that's 45 participants, 23 of whom were on active drug. Of the Healy program, we have 60 people that were on 30 milligram. So taken together, that's about 80 individuals. Yet in the EAP program, we have 180. So 3 times Healy, 2 times our total set. And so that's why this neurofilament understanding from the pre-specified statistical analysis plan the agency asked us to do to seek concordance is exactly that issue. Let's make sure there's not a false positive in your neurofilament benefit. And then let's see if that correlates to survival. And then let's talk about a new drug application. So I think the agency is being wise about this and being careful to use the data that we have. And that data includes the NIH EAP.
Thank you. You know, we've had this asked a couple different ways, and it's kind of a simple, straightforward one. So is this drug basically making your NAD positive level go up? Is it basically making the level go up, and do you clinically measure those levels, the NAD plus levels? And what's the difficulty in doing so? Is it difficult to do so?
Maybe I can start with this one. So essentially, yeah, we are increasing NAD concentrations. We're using the natural bodies of NADH that we have, the natural pools, if you will. And with CNM-Au8, we're taking advantage of catalysis, which happens all the time in our system, and shifting the concentration of NADH to NAD. So yes, the answer is we do. And we have seen that in the MS and Parkinson's disease population, where we've been able to do specific target engagement. It's very hard and almost impossible to do it for those that have ALS because of the mechanism of the analyses. But yes.
And there's also a fair amount of work, Christian, that we've done with induced pluripotent stem cells from ALS individuals who donated their ALS tissue that we then created induced pluripotent stem cells from. And we can look specifically at NAD and see the increase there mechanistically. And so since the mitochondria that's fighting back, as it were, against this ALS assault is so remarkably energetic dependent, and it needs the energy to basically continue doing its core function, that's where NAD as well as ATP becomes pretty relevant for the way an electron exchange is occurring with a gold nanocatalyst like we have here with Au8.
Thank you. Got time for one or two more questions. Adia, did you have one lined up?
Yeah, the question I lined up kind of got answered, but I feel like it'd be good to cover. Why not neurofilament heavy chain if the CNM is crossing the blood-brain barrier?
So, Marjan, do you have any comment on neurofilament heavy?
It's not something that we've looked at, so it's hard for me to comment
Yeah,
on.
we've been very focused on neurofilament light, mainly because, and by the way, we've done both serum and plasma with neurofilament light. Healy, as you maybe well remember, pre-specified serum, and we pre-specified plasma. So we have both of those data sets. But in both cases, our focus has been neurofilament light. And yes, we do cross the blood-brain barrier, but even Dr. Bowser and his work with us has pretty been focused on light. So heavy remains an area of research that we couldn't explore.
And obviously, we're interested in, but it's not information that we currently have as it pertains to CNM-Au8.
And I should also add just one little point more. Quanterix, who does all of this work for us at the laboratory, their plate-to-plate assessments has been pretty much centered on neurofilament light assessment thus far.
Thank you, guys. And we're at the end of the time. So if Christian, you want to...
Yeah, I was going to ask one more. which I think is an important one because so many people are testing different therapies. They're on edaravone and others. So CNM acts as a redox regulator. Edaravone is also a redox regulator. Do you think the people who took edaravone and CNM could have a synergistic effect, or did you account for any difference between the groups of patients containing those exclusively on CNM-Au8 versus those on edaravone as well?
So head to head, we don't have the data to say so. We have allowed background therapy within the programs on stable therapy. So presumably CNM-Au8 is thought to be add-on therapy, whether it was for prior programs or as we move into the phase 3. But head to head, we don't have the information.
Yeah, we'd be guessing a bit, but I think there is a point to be made here, even because there's some supplements questions in the chat. CNM-Au8 is not hepatically metabolized. It is leaving the body unchanged after it does its job. So that's the reason we don't have any concern about drug-drug interactions. In all of our clinical programs, both in Parkinson's, MS, and especially in ALS, we ask the people who want to come into a clinical study to take whatever drug they want. We have not actually precluded other meds, except in the case of the EAP, where there's some regulatory reasons why they cannot be on new advanced therapies. But we've actually put our drug on top of anything. So even if the clinician wants to increase my dose with something, that's fine. Clene doesn't have a challenge with this. It becomes entirely a clinical call. And also to the point, Christian, yes, there is some interesting redox synergies that probably could be had or argued with a couple different things, including in the supplement category, but we've not studied this in particular, so we would be guessing a bit and shouldn't probably do that.
Thank you so much. As a closing for you, in terms of qualifying to enroll or learning more about the phase 3 protocol for inclusion and exclusion criteria, where can people learn more?
Yeah, I mean, we'll definitely have more information as we start to launch within the areas. We are working on developing a website where folks can reach out to us. In the meantime, you can directly reach out to us at Clene, and we'll definitely take your information and sort of create a list so that at least when the websites become available with additional information and as sites come open across the world, we'll be able to post that as well as updates, enrollment criteria, inclusion, exclusion criteria. So we are working on that.
And we'll be back to have you when you have more information as well. So thank you so much. Thanks for all your time. Thank you to the community for some really great questions. I'm sorry we didn't get to all of them. There were a lot of questions. So I know Rob and Marjan are both great at follow-ups with us. So we'll compile the list and see what we can do to follow up and send additional responses back in a newsletter or just have you back soon.
Thank you so much for the invitation.
Thank you, guys.
Thank you.
Well,
Well, there you have it, everyone. Brought right to your living room. I just want to say thank you to those presenters tonight and to each one of you, because when you show up, we know we're doing a good job. I have an ask, and I need some help. So it's kind of interesting. We're looking for a new team that would be willing to do interviews. An example, NBC, CBS, Newsweek. I'm looking for a group who are doing our studies and who would be feeling comfortable because we've had a few interviews so far, and we know that the interviewer is very conscious of ALS and is very supportive. So if you're a little nervous about being on television or in Newsweek, that's okay. It's totally normal. But if you'd like to join me, please send me an email at mcfinn@everythingals.org, and I will get back to you and talk to you more about these possibilities. That's my one ask for today. My other ask is, hi, everybody. Welcome to the program. I'm so glad that we got together. And always remember, it's your program. And so the things that you found of interest tonight that you might like to say a few words about, that'd be great if you'd like.